Yes, welcome back, friends. I've seen a very great confusion among students in confusing different parts of the topic of genetics, and more especially in the parts of evidence and even some other parts of protein synthesis. So for this time, I don't have the time for recording the video for the whole topic, but I can record some few parts which confuse many students so that to help raising your performance. Now, in today's video, I will go to explain about the evidence that hereditary material are located in the nucleus. And for the today's session, I will explain the evidence from green algae. Sometimes it's called the hammering experiment or hammering acetabularia grafting experiment. So I'll be using this Word document. Also I'll be using my PowerPoints, which I will show them later. But all in all, my aim is for you to understand this part and to be easy for you to memorize during your examination. Because I know this is one among the parts in genetics which are very difficult to be memorized if you don't know the history of this hammering experiment, how it was performed. And if you don't know the history, that means to be very difficult for you to memorize it. Now, let's go together. It is called the evidence from green algae because the species of a microorganism or the algae which was used, it was the green algae. And the genus of the algae used, it was acetabularia. So Hamelin, he was a scientist who performed this experiment. By his full name, he is called as Joachim Hamelin. By his full name, he is Joachim Hamelin, and he was a pioneer Danish-German botanist founded by Nazi German. He was founded by Nazi German, and he determined that the nucleus of a cell controlled the development of an organism. So, he is the guy who anaitwa Joachim Hamelin. So, in 1934, he demonstrated that the nucleus determines the characters of the cell and ultimately the characters of an individual. In other words, the cell stores hereditary material. This is what was determined by Joachim Hamelin. And how he performed the experiment. He performed the experiment by using the unicellular green algae of genus Acetabularia, which grow up to 6 centimeters. As model organism for the investigation. So alitumia green algae of genus Acetabularia to demonstrate the experiment. Now the individual algae consists of 3 parts: distinct foot that contain nucleus, stalk, and cup region. And this is the largest unicellular organism. Now before explaining this diagram, let me go back to my Word document and explain what or how he performed the experiment. Now we are saying that what Hamelin did, he proved that the cell stores hereditary material in the nucleus. Now the experiment was done by Danish botanist Joachim Hamelin. Some of them, they are saying in the German. It is because, as how I told you in the PowerPoint, that he was the Danish-German biologist. Now, by using unicellular green algae, they have only one cell which is located in the foot region. Unicellular green algae of genus Acetabularia, which grow up to 6 centimeters as model organism for investigation. Now, what he did, or how he performed the experiment. Individual algae consists of 3 parts, as we said: a distinct foot that contains nucleus, a stalk, and the cup region. Foot, stalk, and the cup region. The upper cup or crown has a characteristic shape for each species. So, for example, here in the diagram, the cup of this species is different from the cup of this species. However, both of these, they are genus Acetabularia. But this one is Acetabularia caneolata, and this one is Acetabularia mediterranea. Now, what you are saying is that the cup or crown has a characteristic shape for each species. Cup has regeneration potential if damaged or removed. The single nucleus is seen in rhizoid region or foot region. So this is a unicellular organism, and the nucleus is located in the foot region of this unicellular organism. Now we use the two different species of Acetabularia, which differ in shape of their cups. Acetabularia mediterranea with a characteristic cup of 81 rays and rounded tips. 81 rays and rounded tips. This is Acetabularia mediterranea. As you can see, there are many rays of the cup. And then Acetabularia crenulata with a cup of 31 rays and pointed tips. 31 rays and pointed tips. If this diagram did not show you clearly the difference between these two species of Acetabularia, let's see this diagram. It is somehow explain how they are different. This is Acetabularia mediterranea, and it has 81 rays with rounded tips, while the Acetabularia crenulata, it has 31 rays with pointed tips. So you can observe the difference in the cups between these two species of Acetabularia. Now, the algae are capable of regeneration from any part. They are capable of regeneration from any part. So that cup, stalk, or even nucleated rhizoid portion is removed. It will soon regenerate and give rise to a wild plant. So if any part of the plant it is removed, any part of the plant is removed, it can be regenerated. Any part, if it is whether the cup, the stalk, or even the nucleated rhizoid portion is removed, it will soon regenerate and give rise to the whole plant. Now you must understand this very important concept. But we are saying that but the enucleated portion, enucleated, that means it does not contain nucleus. Enucleated, that means it lack. Enucleated portion on continuous decapitation or removal loses the regeneration capacity, whereas the nucleated region always retains the regeneration capacity. So maeneo ambayo yanakuwa hayana nucleus, that means ni stalk pamoja na nani na cape. Stalk na cape hazina nucleus. So on continuous decapitation, on continuous decapitation, unavyozidi kukata mara nyingi, hizi zina lose their ability to regenerate into a full plant. So they lose their ability to regenerate into the full plant. Now, the part of the Acetabularia which contain nucleus, it retains its regeneration capacity. But the nucleated region always retains the regeneration capacity. The nucleated region always retain the regeneration capacity. So nucleated region ambayo ni rhizoid au foot inakuwa ina retain regeneration capacity. Now by amputating or grafting, that's why it is called hammering grafting experiment. Amputation, that means cutting. So you are cutting a certain part, then you put it on another part. That's what you call as amputation. So unakata one species, then you put it on another species. By amputating different areas, grafting to other algae or another species, and seeing what part would grow, he observed that the form that was found in the foot region is the one that would influence new growth in the cup region. The form that was found in the foot region is the one that would influence new growth in the cup region. So if the foot region contains the nucleus of Acetabularia mediterranea, also the cup region would contain the cup of Acetabularia mediterranea. Vice versa is also true. If the foot region contains the nucleus of Acetabularia crenulata, the cup region also will contain nucleus of Acetabularia crenulata. Now he observed that the form that was found in the foot region, the one that would influence the new growth in the cup region. This led Hammerling to hypothesize that hereditary information driving new growth or regeneration of the cup is found within the foot of acetabularia and possibly in the nucleus. This is how Hammerling experiment ends. Unaweza kuona aligrift hapa akaweka pale, hii cup akatupa pembeni, hii cup akatupa pembeni. So unaona hii cup alitupa akaja grift akaweka hapa, lakini kichokuja grow kujuu kilifanana na cha chini. Why? It is because this nucleus is the one which control the growth of this one, regeneration of this one. Again, alivyo grift kwa akaweka huko, cup ya hapa juu akatupa, development ikatokea vile. So nucleus in the base determines the type of the cup regenerated. Or you can see in this diagram, unaona he grifted the cup and then akatupa, akagrift akachukua hii akajoin na stock yule pale, nucleus yule pale akaachana naye, yule pale kepo akatupa pembeni. Unaona pale kwanza walitengeneza intermediate type, manake ni kama huyu na huyu alichangiana, then walivukata tena ile wakatupa, hiyo kuja grow final ni ipi? Ni huku. So that means nucleus hapa chini ndio ambayo ina control hiyo development. Again, you can see from these diagrams, they are the same, but you can see kama nucleus ikiwa mixed itatokea intermediate type like this one, intermediate type. But nucleus kigrift hata kama kuchukua stock ya kule ka mix up, stock ya kuka mix up utapata intermediate, lakini baadaye ukifanya decapitation, ndio maana tumekwambia hapa only continuous tumesemaje, the algae are capable of regeneration from any part, so that cup, stalk, or even nucleated rhizode portion is removed. It will soon regenerate and give rise to the whole plant. But in nucleated portion, only continuous decapitation. This is what we are saying. Only continuous decapitation. Yani umegrift, umepata ambaye ni intermediate, umedecapitate tena, umekata tena, umeondoa, unafanya process repeated. Ukikata tena kuta amrudi tena kule mwanzoni. On continuous decapitation, we are saying that on continuous decapitation or removal loses regeneration capacity. Unaona kwa hapa tufanya continuous, huyo hapa alikosa regeneration capacity, so tukapata ile wa mwanzo, but the nucleated part continues. So loses the regeneration capacity, where the nucleated region always retains the regeneration capacity. So this is how we can conclude the Hammerling experiment.